Hi, hi everyone. Hi everyone. So I am Alejandro Moure Abelenda. I'm coming from Spain, and I would like to tell you a little bit about the technology that we have been developing. Okay, about extraction of ammonium bicarbonate from anaerobic digestate and heterolactic fermentation of dairy slurry. Okay, so we developed these technologies at two different scales. We are talking on lab and commercial scale. So before going deeper in my discussion, I would like to tell you a little bit of the background so everybody understands what I'm talking about, okay? So eventually we produce agricultural waste, manure, and slurry, okay? So traditionally we apply this material directly to land, right? And we can eventually use some additives to improve the properties of these materials during land application, okay? So we are talking about making better fertilizer, minimizing ammonia volatilization, leaching of phosphate, okay? So that is if we have a budget limited, but if we have a greater budget, we can go for investment in an anaerobic digestion plant, right? So in this anaerobic digestion plant, we can be able to start biogas and produce energy. During this fermentation, we have the mineralization, so organic forms of the nutrients goes to ammoniacal nitrogen, for instance. Organic nitrogen goes to ammoniacal nitrogen. So it's in this context when we can think about the fractionation and extraction of the nutrients, because if we apply this at once, the roots of the plant will not be able to grab all the nutrients, right? So here is the when I start to talk about the extraction of ammonium bicarbonate, okay? So I study like three different types of digestate: food waste digestate, agro waste digestate, and ammonium digestate, right? So the food waste, as we've been discussing this morning with Brett, is very interesting because it has a lot of content of ammoniacal nitrogen. Because of the conversion from proteins, all right? So agro waste digestate is more challenging because the content of ammoniacal nitrogen is lower, so it's more difficult to get the extraction of ammonium bicarbonate. And then we have the manure digestate, which is very interesting because of the urine that is excreted by animals. It has a lot of urea, right? So we kind of perform a distillation process to extract this ammonium bicarbonate, right? So if we think a distillation, we need to be aware that both carbon dioxide and ammonia are generally regarded as gases, not condensable vapor under normal conditions, right? So, and if we want to perform this desorption, absorption, and deposition in the conventional distillation setup, we kind of make some assumptions to look whether this equipment can help us. So we did the mechanical methods with some assumptions. Okay, we took some equilibrium data. We see 3 stages, and we say, okay, more or less it's fine. So a total reflux, minimum number of stages, more or less is fine. We are using a Levine condenser connected to the reboiler, which is a round bottom flask where we feed the digestate, and it's connected through the thermometer valve. Important, the thermometer valve is in the neck that connects the reboiler and the condenser because it's in close contact with the gases going upwards, and it gives us the best reading of the temperature, okay? So now here, right, so to follow the extension of this extraction, I also developed a titration method, okay? So the validation of this titration method was done with empirical data. So I determined the content of nitrogen and carbon in the digestate. I performed the titrations, empirical titrations of this digestate. I performed the empirical titrations of model solutions of ammonium bicarbonate, and I also consider theoretical data for the titrations. So what we can see here is as the content of ammonium bicarbonate increases, the steep of the slope is lower, meaning that we need more acid or base to change the pH. And this makes sense because we know that ammonium bicarbonate is one of the main responsible for the fat buffer capacity in the digestate, along with the volatile fatty acid. So now I think, come on, I have a video here, and hopefully it will work. Come on, man, you can do it. You can do it. Come on, hey, come on, it's working. Come on, go, go, go. Oh man. Anyways, let's go back. Yeah, this is one. This is good one. Anyways, so the point I report these videos in the article because I was discussing before with arrogant reviewers, so no one can tell me this was not going to happen, and it was very difficult to replicate the conditions. So the point of these videos is telling the readers of the article how difficult it is to replicate the conditions. So I report the three replicas. You can see here how the form is increasing, right? And if you have to operate at very low temperature, lower than 60 degrees, and very low heating rate. Otherwise, the foam will reach the crystals that are forming around the bulb of the thermometer and will contaminate. So the total operation were 8 hours of heating, like lower than 60 degrees, and the characterization of the product was done overnight, right? So we can see here more how, the deposition of the crystals. You see the deposition of the crystals. So there are more and more crystals every time. So maybe if I press, you can see even through the Levine condenser, more and more crystals, right? So let's move on. Let's move on. Come on. Let me press out. Come on. To replicate. Come on, come on.
So you can take your time.
No, but so these are the final results. To sum up, we have the 3 replicable record. No one is going to tell me this is not going to happen. So we are comparing the initial digestate. The blue line is the initial digestate to the depleted digestate after the distillation, and we see that the depleted digestate has less buffer capacity. The slope of the titration curve is more steep. So we need less acid or less base to reach the titration endpoint, because they contain less ammonium bicarbonate. Why? Because we got the solid ammonium bicarbonate. So based on the mass balance, we start 80% of the ammonium bicarbonate in the food waste digestate. And which kind of ammonium bicarbonate are we extracting? We compare the peaks of FTIR, the absorbance peak of the infrared, to a reagent grade ammonium bicarbonate. And we see that we got exactly the same peaks, with the difference that the height of the peaks is greater. The ammonium bicarbonate have greater peaks than the commercial ammonium bicarbonate. In absorbance, when we have greater peaks, this implies greater concentration, but we cannot have a greater concentration here because it's pure ammonium bicarbonate. So those greater peaks should imply lower stability and greater availability of the nutrients to the plant. And we can discuss this later because people understand differently. For me, I'm talking about lower stability, greater availability to plant. These nutrients are more available, are more likely to be lost, and hopefully trapped by plants. And this is confirmed because when I vacuum dry the replicas, the peaks decrease. We keep the same peaks, but the height of the peaks decrease. So this means that only the crystals more slow release, stable, are able to prevail, and the others go away. So these are more strategies that I've been testing. I have a following video. For instance, here I'm looking to a 70 degrees deposterization. I'm looking to harvest these crystals with a circulating system with the agro-waste digestate, which is more difficult. Or even here, I am using the half depleting the food waste digestate, and then adding an anti-foam, this is another thing, adding an anti-foam K989, so I can increase more the heating and completely deplete the ammonium bicarbonate. So these are different strategies that I've been using. This is another video. This is not really important. This is just to show you how I do the periodical harvesting. So just swapping the thermometer. I'm telling you, this is not really important. The important is the following. Come on. The important is the following part. When I try, Jonas, come, this is important. This is really important. When I was thinking, how I'm going to replicate this at a large scale? how I'm going to replicate this at a larger scale. And I realized that in the biogas pipelines, there was a condensate. In every single anaerobic digestion plant, in the pipelines, there is a condensate. Ladies and gentlemen, please be aware that the water scrubbing is the technology most widely used for upgrading of the biogas to biomethane. Water scrubbing. So let me show you. This is the schematic of the manure digestate plant. So this is the biodigester under mesophilic condition at 40 degrees. This biodigester is fed with dairy, slurry, manure, and whey from cheese making, and other stuff. At the very top, we have a biogas pipeline coming out. In that pipeline, there is condensation. And we have 2 points of sample that I am showing you here. In every single anaerobic digestion plant, this is happening. And we can harvest the condensation at this point. I'm showing you this as a sample, but I actually harvest that condensate, biogas pipeline condensate, in food waste digestate plant, in the manure digestate plant that I just showed you, and in another waste digestate plant. And I compare the amount of ammonium bicarbonate in that condensate with the titration method. I'm going to show you in the next slide how I compare again with different methods. So what we see here, interestingly, and makes sense, we see here that the most saturated aqueous solution in ammonium bicarbonate is that provided by the pasteurizer of the food waste digestate. Food waste digestate, we know it has a lot of ammonia, so it makes sense that CO2 and ammonia meet each other in the gas phase. And it also makes sense that the pasteurizer at 70 degrees enhance the mass transfer and the volatilization of these compounds afterwards. The other interesting part, if we look, we see that the manure digestate, the condensate from the manure digestate pipeline presents more ammonium bicarbonate than the condensate of the food waste digestate. So this means that the amount of ammoniacal nitrogen in the manure digestate is greater than in the food digestate. And by the way, I did the dilution of the samples of condensate because I got a oily phase in the sample. In the sample of the pasteurizer, I got an oily phase, and I did the titration in case these were the volatile fatty acids. Okay, so the titration, it has minimum buffer capacity. So I also estimate the content of ammonium bicarbonate by directly determining, not only with the titration methodology, but with the TOC, directly determining the content of total nitrogen and total carbon, which is inorganic, because which other form is going to be in the aqueous solution. And I empirically precipitate and extract this material using an anti-solvent method, okay? I'll cool down at 2 degrees the aqueous solution, and I add acetone, which is the anti-solvent. So here we can see that there is the direct relation, okay? The same trend we found. And the blue trend is the minimum amount of acetone that I need to add to promote this supersaturation, as I'm going to show you. Come on, this video has to work. So in this video, I am showing you how I promote this the supersaturation. So I have 3 milliliters of the condensate at 2 degrees, and I start acetone in a small step. And you see that it starts to become whitish. You see? And I keep adding, and I take note of that. This is the minimum amount that I need to promote the supersaturation. And then I add until 40 milliliters to recover as much as possible, as I did with all samples. And the same results as before. The ammonium bicarbonate that was isolated, come on, man. The ammonium bicarbonate that was isolated has greater peaks than the commercial fertilizer. Only here the peaks were small, because when few ammonium bicarbonate is produced, only those crystals that present higher stability prevail. If you produce a lot, it goes with there easily. So now that's all I have to say related to ammonium bicarbonate. I did not know that here you use it as fumigation. I know that in Europe we don't. It's not so far considered a fertilizer. Okay, and in the last conference I've been in Europe, we were discussing this point. So now let me tell you a little bit about a technology that I've been developing, which is the heterolactic fermentation of the dairy slurry. Okay, it's my business. I'm doing this not for a living, but so why? This is how heterolactic fermentation occurs, okay? This is a schematic of the lagoon that I will show you in a later. The lagoon that I was talking before. So here at the bottom, we have the manure, the solid manure, submerged, completely submerged in water under anaerobic conditions. And in that soaking process, the hydrolysis of the fibers take place. And that generates fermentable sugars. Here I have a molecule of glucose. And microbes are able to break that molecules of glucose in a molecule of lactic acid, ethanol, and carbon dioxide. This is heterolactic. If where homolactic would be two molecules of glucose, which by the way is nothing new. This is what is done for the preparation of silages with corn and grass for grasses. That is heterolactic fermentation. This graph I prepared for our outreach event for telling young people that in the same way we provide our body with food in a good state to grow our muscles, we need to provide the soil with a material in a good state, fermented material, and not putrefying material, right? So, related to the homolactic fermentation, when you ask farmers whether they use something to promote this in silages, some of them, they tell you, I am using propionic, butyric, formic acid as preservant. Other tells you, I am using lactic acid bacteria as inoculum. Others say, I am following my own criteria. This is what I find out. I am originally from the northwest of Spain. The gentleman is from here, from Cordoba. I am from the northwest of Spain, near Santiago de Compostela. I am from a village nearby. And I am moving around. I am going to Mazarico, Santa Comba. I am going to Trazo. I am going to a different county. I am going to Friol, a different county, Silleda. And I am collaborating, building business with farmers. I am telling them to ferment their manure. And what I'm going to show you is an example of how we do it. Why these people want to improve, they do the fermentation of their manure. So this is their lagoon. And this gentleman, by the way, this is a lagoon of 2 million liters. It's not a joke. And this gentleman wants to put this pile of cow bedding that we were just talking. This is a straw. This cow bedding is used for calves and dry cows. And if you want, I can explain you later what is the logistics in the farm, right? Because otherwise, if you use this hot bedding, this is hot bedding, they will have the mastitis, right? So this gentleman wants to put this pile in the lagoon. Why he wants to put this pile in the lagoon? For a number of reasons, but mainly because of the policymakers that they are telling farmers to build another facility of 10,000 euros or more to store this material. And the farmer says, well, I prefer to pour it in the lagoon, which also helps the farmer afterwards in fertilization, because it does improve fertilization. But you know, it's not always positive. Sometimes the fermentation of the heterolactic fermentation is inconvenient for them. And here we have to come just before, maybe that was the reason for the delay of my flight. Just before coming here, I have to go to this farm to add a controller for this fermentation, because the foam was reaching to the cow, to the feet of the cows. And this could have a problem. This could have a problem in producing irritation in the hooves of the cow. You see, dermatitis is called, you see. So I have to go there, and I think I just received the payment. So I think it went good. So just to finalize my presentation, these are the top reasons I find out why farmers want to ferment their manure. First of all, they want an homogeneous fluid. We were discussing here he said here in the US they want liquid fertilizer. Well, there is not that much difference. They want homogeneous fluid. Sometimes they cannot put the stirrer, or sometimes if they put it, they have to stir it a lot to take it out. So this is one. Other is that milk factories like Nestle, Lactalis, which is a friend company, currently they demand more and more this kind of technologies. Okay, they ask the farmer whether they apply these cocktails, all right. This might be because they want to decrease their carbon footprint. They care about the greenhouse gas. Farmers may care, but they care more about their pocket as well. It's true. And what do they care is about the smell, which Brett was telling us. Brett was telling us. And they tell me as well, I have to do something with this manure because neighbors are complaining and I don't want to have problems with the neighbors. So you are going to tell me how this helps. Well, you got to think that this is an anaerobic fermentation but does not produce methane. We can discuss later if we feed this to anaerobic digestion, whether it increases the biogas production, and it does. In Ramiran conference that I went, people advocating for this technology confirm that. And you talk to farmers, they say also greater crops. At the end of the day, is what farmers says, you know. And when we discuss about the price, they always tell me the same. If the product that you are bringing here does what you are telling us, then it's not expensive. No problem at all. Yeah, that's all. I am supported by Spanish government for the ammonium bicarbonate with the British government and also my own initiative. That's all in my presentation.